due to sudden off my PC and internet. Okay, please share again. And just waiting for other participant. Just give me a 1 or 2 minutes so that all can join. Okay, I think only you or Mariam Rajput. Okay, no, please perform next step. Other participant can watch a recording for next step. Okay, no, you have selected DESeq. No, within inverted comma. Within bracket, remove inverted comma. Type count data, count, C capital, C small. Select this function, count data, equal as dot matrix. It's mean we want to convert our count data into matrix as dot matrix. As dot, no, not dot, dot mean point. Yes, point as point matrix. Select this function, first one. This function will convert your count data into matrix and give your count data name. Your file name is counts. In line number 12, you have given name counts to your data. You can also check in screen number 3, your counts data file name is no counts. So within bracket type counts. Yes. Okay. I'll come to bracket. After two brackets, add comma. Press enter button. No. After comma, press enter button. Type after comma type call data, call, select call data, yes. No, come to start of call data, click on before C, yes, and press enter button. Why it's, I think you have add a space after bracket. It should be a remove space, yes. Again, comma, and run this line. Sorry, and break this line. Yes, no, it's good. Okay. Now next step is it's called data, not function. Call data, it give you equal sign. Remove and type it again. Remove call, remove brackets. Please remove brackets. Also remove data. Again type data. It will give you suggestion. Yes, select first option, data equal. Yes. Meta. No, your column data equal to metadata. Meta. Your file name is meta. Select your file name is meta. After meta, add comma and press enter button. Yes. No type. DSign. DETI. Sorry, DESI. Design. Select this one. Equal tilde sign, or you can say no sign. I am sending this sign in group. Zoom chat you can copy and use. Please check this sign. Copy and use. Equals type cell. After type cell, CELL cell plus DEX. Cell. After cell add plus and type DEX. DEX. Manually type DEX and close bracket. Okay. Now run this line. It will convert your data into DESeq object. Now give a name to this results. Come to line number 21. Add less than minus sign and give a name DDS. Yes. And run this line. Now you can check data appeared in screen number 3. DDS. Now your board data are in single files and is converted into DESeq object. Now next step is you need to remove low quality read. So come to next step, give heading, line number 25. Remove low quality reads. Okay, now our file name is DDS. So type your file name DDS. And add square bracket. After DDS, add square bracket. Within square bracket, type row sums, row sum, S is capital SUMS, row SUMS, sums, again bracket, small bracket, counts, within small bracket, please. Open bracket, first open then close, not close. After sum, yes, open and close. No, within these brackets, type counts. Count is your file name, and again type bracket after counts. After counts, within bracket, again add opening and closing bracket. Yes, and type, no, don't close after count. Don't close after count. Yes, yes. And type here DDS, your object name. Now come to after bracket, after two brackets, or two small brackets. Yes, add here greater sign, greater than 1. After add comma, it's mean if your this script mean remove all values which are less than 1 and retain all values which are greater than 1. So if you have low quality reads, only by chance you get some genes, you can remove, because these genes create problem for your actual showing your actual results, and these are by chance or by mistakes, so have low qualities. Now run this line. It will remove low quality reads from your data. Now you can also give name DDS. So again name it's DDS. No, line number 26. Give less than minus sign and name DDS. It's mean we have applied changes in the file DDS, and after applying changes, its again name is DDS. Now run, and now run this line again, line number 26. Okay. Now you can increase or decrease the number of low quality reads. If you adjust 10, it is high or good quality. If you check add 10 instead of 1, and run, it's mean there is no any under values which are 10. Okay. No alert 1, because it will show you a maximum outputs. Now we have file which have high quality data, and we have converted our data into a matrix, or you can say DESeq object. In our next class, we will perform differential gene expression analysis and some other steps for preparing for next analysis. Okay, thank you. Okay, Mariam Rajput, if you don't have DESeq object to first install this tool. In our yesterday class, we have learned how to install this DESeq2 package. So before next class, install this tool. Okay, thank you. Allah Hafiz.